Thank you all for joining us today. I'm Pat Furlong, and president of PPMD, and we're really delighted that you could join us. Today we're going to focus on Wave Life Sciences and their technology moving forward into Duchenne muscular dystrophy. We're joined today by Wendy Erler. I think many of you might know Wendy. She leads their patient advocacy. Joined by Professor Matt Wood. We've known Matt for a very long time, and he's been involved in exon skipping technologies and the chemistry behind them for many, many years. And also Michael Panzera. Dr. Panzera leads the Wave Life Sciences in the Neurology Division. So with that, I'm going to turn this over to Wendy, and we'll begin the webinar. Wendy?
Thank you, Pat. Hello to everyone, and a special thank you to PPMD for hosting today, and to all of you for joining us. We're excited to introduce you to our company, Wave Life Sciences, and to our work in Duchenne. This is our agenda for today. I'm going to do a brief introduction, and then Matt Wood will go through development of Stereopure ASOs in DMD, followed by Mike reviewing our program in particular, and then we'll have time for Q&A. Our forward-looking statements. On this slide, we acknowledge that in this presentation we use forward-looking statements often identified by words such as may, will, should, plan, potential, or others, or even the negative of these terms or other similar expressions. These statements in this presentation are only predictions, as we are still in the clinical development stage and just starting our clinical trials. We have based these forward-looking statements largely on our current expectations and projections about future events. In other words, there's a lot of hope and promise on this slide. I'm Wendy Erler, and I lead patient advocacy at Wave. I joined Wave about 2 years ago and have been working with the Duchenne community throughout the process as we have moved from early preclinical to the clinical stage of our DMD development program. Last week, we announced the initiation of our first clinical trial, which was planned with input from the Duchenne community. And thank you to everyone who's been involved with us already. We will continue to work closely with the community as we advance our exon skipping programs. Our expertise and core focus is in neurology, and we are developing targeted therapies for patients impacted by neurological rare diseases, particularly genetic diseases. In addition to initiating a clinical trial in Duchenne, we have 2 clinical trials in Huntington's disease and planned clinical programs in ALS and FTD. The foundational approach to treating these diseases is the same. We build rationally designed stereopure nucleic acid therapeutics utilizing multiple modalities, including exon skipping. At the bottom of the slide, you see we have 6 proprietary neurology development programs planned by the end of 2018, and very excited about having opened our manufacturing facility in Lexington, Massachusetts. Understanding that actually making drugs for DMD is complex and challenging, we made a decision early on to invest in manufacturing capability and infrastructure. We recently opened our 90,000 square feet manufacturing facility, which gives us the capability to control the supply chain and ensure drug supply. This site is our second in the U.S. with our corporate headquarters in Cambridge, Massachusetts. This gives you a little bit of an overview of the timeline of Wave. In 2013, 2 companies merged to own everything from manufacturing, design, and optimization all the way through to developing drugs. These 2 companies were Dr. Wada's CRO group in Japan and Greg Verdine's early-stage company called Ontori. Here's a little Wave trivia. The WA in Wave comes from Dr. Wada and the VE from Dr. Verdine. After the 2 companies merged, Paul Bolno joined as president and CEO. Chemistry is the true foundation of Wave and makes me wish I paid more attention in high school. Merging the 2 companies under one umbrella gave us one company that owns everything: design, optimization, and drug development. Wave became a public company at the end of 2015 and had a big year in 2016 with a partnership with Pfizer to explore our technology specifically in hepatic targets. While we focus internally on neuromuscular and other neurology, importantly to this audience, we've named follow-on candidates beyond exon 51 in exon 53. This year has been busy as well. As I mentioned earlier, we've initiated 3 clinical trials, opened our manufacturing facility, and announced our next DMD clinical development program. This This is a snapshot of WAVE at a glance. We're the only company and the first company working to develop Stereopure drugs. We have multiple programs in drug development, while at the same time we're continuing to work in drug discovery to see where else we can apply stereochemistry to make a meaningful difference in challenging diseases. On this slide, you see a snapshot of just our DMD pipeline. As I mentioned, we've advanced our exon 51 skipping programs to the clinic, and we've announced our development work in exon 53. We have other research projects exploring additional exons. You will learn more about our exon skipping approach in today's webinar. I'm delighted to introduce you to Matt Wood. There's no one better to explain exon skipping and the impact of stereopure chemistry than Dr. Wood. Matthew Wood is a professor of neuroscience at Oxford University and has been working with WAVE on our Duchenne work from the early days. Thank you, Matt.
Thank you, Wendy. Hello, everyone. As Wendy said, I've been involved working with WAVE on their program, and more broadly, I've been involved in working on exon skipping therapies for pretty much the last 15 years, covering most of the period of the exciting development of these therapies for Duchenne. And what I'm planning to do is to try and give an overview of exon skipping therapies, but also to put the exciting developments that WAVE is pursuing into a broader context. So on this slide, I wanted just to give you a historical perspective of where we've come from and hopefully where we're going to in exon skipping therapies. Many of you will be aware that Duchenne was first described pretty much 150 years ago, and there was a very important decade in the 1980s when we began to understand more precisely exactly what causes Duchenne. The dystrophin gene was identified, and the protein produced by that gene was studied, and we now understand a great deal about how it functions. But it was quite a wait from then until the early 2000s when the very first work on exon skipping began to make an impact. And in fact, my own group and research began in about 2003, at the beginning of this period. The first exon skipping therapies were in development in the early 2000s, and the first clinical trials began around about 2007, pretty much a decade ago. And this period through from 2007 to where we are now in 2017, I would characterize as the period during which the first generation of exon skipping drugs have been developed. And these are very much classical oligonucleotide therapies, as I'll describe. There was a period around 2012 to 2014 when there was a lot of activity and multiple trials in progress being developed by Crescenza BioMarin, but also by Sarepta. And ultimately, the drug being developed by Crescenza BioMarin, drisapersen, was not approved. However, a major breakthrough in 2016 occurred, and you'll all be aware of this, when eteplirsen, the Sarepta drug, was approved as the first exon-skipping therapy for Duchenne. And more recently, towards the end of this year, WAVE have announced the initiation of their clinical program. And so this period, the last 10 years or so, really is the period of first-generation traditional exon-skipping therapies. But this is now opening the door in 2017 to a new generation, or the next generation of therapies, which are going to generate drugs which we hope are going to have much better activity and benefit for patients in the future. Now, to take a step backwards and to say something about DMD and how exon skipping works in the case of Duchenne. DMD, as you'll all be aware, is a genetic disorder. It's inherited. And the gene that is responsible for causing this disorder was discovered in the 1980s, as I've described, and is the DMD gene. And it produces a protein called dystrophin. And dystrophin is what we call a structural protein, and it plays an absolutely essential role in muscles. And a schematic picture of a muscle with all the muscle fibers is shown in this slide. And dystrophin acts to stabilize muscles during their contractile function. And it acts as a kind of glue, holding the muscles together and enabling them to function even under high stress during activity. Without dystrophin, which is the case in Duchenne, these cells become damaged progressively over time, and the muscles weaken and degenerate. And this is really the major consequence of not having dystrophin, although there are consequences in other tissues too, such as the heart and heart muscle and the brain and the nervous system. So what is the dystrophin gene? Well, genes are located on chromosomes that are present in most of our cells, and the genes themselves comprise DNA, which you'll all have heard about. And the DNA itself is made up of what we call nucleotide bases. And this is where the DNA code is written. And these bases are essentially of four types shown here, A, G, C, and T. And along the stretch of DNA, the sequence of these bases or sequence of these letters generates the code for a particular gene product, and in the case of our interest today, dystrophin. Now, mutations or errors in the DNA are caused by changes or errors in one or more of these nucleotide bases. In other words, changes to the code, the consequence of which is a disease. Now, the other important concept with respect to DNA and its organization in genes is that DNA within a gene is organized into regions that we call exons, shown here in red, and introns, shown in blue. The exons contain the DNA code for making a protein. And that code is converted into an RNA or a messenger RNA and then ultimately used to make the protein. The blue stretches or introns are what we call non-coding regions, and they interrupt the exons. And so during the production of a protein, such as dystrophin, a very important step is the stitching together of all of these exons and the removal of the introns. And this process is called splicing. The exons are spliced together so that they can then generate the protein of interest, and in our case, we're thinking about dystrophin. So what happens in Duchenne? What goes wrong in DMD? Well, in DMD, the genetic mutations that I've referred to already, the changes in the nucleotide bases, are typically quite large changes that we call deletions. And the consequence of those deletions is typically that what we call the reading frame and the splicing of the exons is disrupted. Now, the reading frame is essentially the code making sense. And so, as a result of the error or the mutation, the genetic code, the sequence of bases, no longer makes readable sense. And this is where the problems arise. So shown here on this slide is an example of just such a deletion to explain this in more detail. So here is a snapshot of the dystrophin gene. And we're looking very specifically at a small number of the exons, shown here, exon 46, 47, 51, 52, and 53, which is in the central part of the dystrophin gene. Now, in this particular case, there has been a mutation between exons 47 and 51, a large mutation, which is quite typical, which we call a deletion, where a long stretch of nucleotide bases has been removed. And the consequence of this is that the reading frame has been disrupted. The code in this region of the gene no longer makes sense, and the splicing, or the stitching together of these exons, is disrupted. And as a consequence of that disrupted reading frame and disrupted splicing, dystrophin protein is not made. And that lack of dystrophin protein is what leads to disease. And so how can this mutation, how can these mutations, these errors in the genetic code in the dystrophin gene, how could they be corrected in order to make dystrophin protein and improve the course of the disease? And the method that's been developed over the last 2 decades is a method called exon skipping. And the exon that the community has focused on a lot and is the target of eteplirsen, the drug developed by Sarepta, is exon 51. Now, exon skipping is designed specifically to correct this abnormal reading frame and enable the production of functional dystrophin protein. And that protein is expected to result in a clinical benefit for patients. So this is essentially how it works. We're looking at pretty much the same image as we had on the previous slide, where there has been a large deletion between exons 47 and 51. And as we know now, this disrupts splicing and no dystrophin protein is made. But what can be done here is to engineer an oligonucleotide, shown in blue, and I'll come on to explain exactly what oligonucleotides are in the next slide, to engineer an oligonucleotide which attaches to 51, exon 51, and blocks or masks recognition of that exon. And so during the process of splicing, when the exons are stitched together, the splicing process skips over or misses out exon 51. And as you can see, exons 47 and 52 fit together very nicely, and that's shown by the way that they're drawn in this schematic cartoon. So by skipping out exon 51, exons 47 and exons 52 now fit together, and we say that the reading frame has been restored. And because the reading frame is restored, this allows the messenger RNA to be stabilized and dystrophin protein to be made. So simply by skipping over a single exon, in this case, the reading frame can be restructured and dystrophin protein is made. So what are these oligonucleotide drugs? Well, oligonucleotides, as the name suggests, is a type of drug which comprises a synthetic molecule that is a short, single-stranded stretch of nucleotide bases, and these are linked together. So a typical oligonucleotide could be about 20 nucleotide bases in length, schematically shown here as a single base here and another base here, and these are linked together through what we call the oligonucleotide backbone in order to create a stretch of nucleotide bases, which we call an oligonucleotide. And the bases, the sequence of these bases, and these are the same A, G, C, and T bases that we referred to earlier, the sequence of these bases in the oligonucleotide allows the targeting of a specific DNA or RNA sequence. In other words, an oligonucleotide can be synthesized to specifically target exon 51 or exon 53. Now, the bad news is that oligonucleotides make poor drugs. However, the good news is that over the last 20 to 30 years, a lot of work has been done to understand how to modify the chemistry of the oligonucleotide to make these compounds much better drugs. So essentially key to the whole process is chemistry. So the oligonucleotides in their native state are typically rapidly degraded and eliminated from the body. But through the application of chemistry, the properties of the oligonucleotides can be improved to make them more stable and more drug-like and much better for therapeutic applications. Now, shown in the schematic image here are the types of chemical modifications that can be made to oligonucleotides to improve their properties. One of the most important types of modifications is to the backbone. In other words, the links between the oligonucleotide bases. But modifications can also be made to the bases, to the sugars, and in fact, changes can be made to either end of the oligonucleotide by attaching them to other types of chemical compounds. So a wide range of chemical changes can be made to improve the properties of these drugs. Now, the traditional or classical approach to developing oligonucleotide therapies, and the one that has really been pursued over the last decade or two, is shown in this slide here. A very common modification to the backbone of oligonucleotides to improve their drug-like properties and improve their stability is the introduction of a linker to the backbone called a phosphorothioate linker or phosphorothioate backbone chemistry. And you can see the difference between the phosphodiester here and the phosphorothioate here, which really is the S or the sulfur atom which is included in this backbone change. Now, this is very important because it has important favorable properties, improved stability, but what it does do is introduce what we call a chiral center. And that's shown here. And this is really a very important point to appreciate because it's highly relevant to all of the technology that WAVE is developing. This phosphorothioate backbone here can exist in two forms: an RP or a right-handed form and an SP or a left-handed form. In other words, two structurally different forms, one of which is a mirror image of the other. Now, typically, when an oligonucleotide is synthesized, and this is shown schematically at the bottom of the slide here, each of the nucleotides is shown in gray, and the linkages are shown as these links between each nucleotide base. Typically, in a phosphorothioate backbone, the links between these are entirely uncontrolled and randomized. So a link could either be an RP link or an SP link, and there's no control over that during the synthesis of these molecules. So whether it is an RP or an SP is entirely random. And this is what is typically true of all first-generation oligonucleotide drugs that have been in development over the last few decades.
So, Matthew, I'm going to stop you there for just a question. If you'll go back to that slide. The community is very familiar with the backbone of the PMO, and more recently hearing of the peptide conjugated or PPMO, and then from the Prosenza BioMarin studies or GSK Prosenza BioMarin studies, the 2-methyl. And so, could you just comment on? You said that you were just talking about the uncontrolled linkage. Are those backbones that we are familiar with, PMO, 2-methyl, et cetera, are they the same in terms of being uncontrolled? And so this addition that you're talking about, the phosphorothioate, makes all of those different, or could you just sort of narrow that down for us?
Yes, of course, Pat. I think the phosphorothioate is a particular example of a backbone modification. And this is highly relevant to the drisapersen compound, but also to the morpholino-eteplirsen compound. Although in the case of morpholino, the backbone linkage is a slightly different chemistry. Now, both of those compounds, the drisapersen and the eteplirsen, have chirality built into the backbone. So, although phosphorothioate is not relevant to both of those drugs, what is true of both of those drugs is that there is chirality or randomness built into the linkages in the backbones of those drugs. So when those molecules are produced, the links, the backbone links between the nucleotides are entirely random and uncontrolled. The sequence, of course, is highly controlled, but the links in the backbone are uncontrolled, and it could either be a right-handed link or a left-handed link in both of those chemistries.
Thank you.
And so, as I've said, chirality is naturally introduced into all of the first-generation drugs. Of course, the phosphorothioates, as I've said, the phosphorothioate backbones were introduced originally because they have very beneficial properties. They provide stability, they provide what we call bioavailability in the tissues of the body, so they improve the drug-like qualities of the oligonucleotide, but they introduce chirality at each linkage along the oligonucleotide backbone. And if the chirality is not controlled, in other words, if it's randomized at each nucleotide junction, this results in a mixture of drugs. So a single preparation will contain a chiral mixture, and that's shown schematically here because you can very easily appreciate from this schematic that the left-handed version of the linkage and the right-handed version are very different. They cannot be superimposed. They're mirror images, but structurally they are completely different. Now, the key to WAVE's technology is the ability to control the orientation of these chiral linkages so that what is produced when an oligonucleotide is made is a pure oligonucleotide, or what is called a stereopure, with the chiral linkages controlled precisely at every nucleotide junction. And this slide really helps to explain that in a little bit more detail. So the traditional classical method is shown on the left-hand side, and the result of this, where the chiral linkages are randomized and not controlled, is the production of a mixture of drug. And you can see from the schematic below that if we're considering an oligonucleotide that is of the order of 20 nucleotides in length, and each of these gray blobs represents a nucleotide, then this results in a mixture of over 500,000 molecules within the drug, a diverse mixture. So rather than being pure, it contains a random mixture, and this may have implications for stability, activity, and safety of the drug. In contrast, WAVE's approach is an approach that one can think of as rational design, being able to precisely control every element of the oligonucleotide. So WAVE is able to control the chirality so that what is produced is a single drug that is a stereopure drug where the chirality is controlled and is exactly the same in every single molecule. And that's shown down below here. So, for example, it could be controlled in such a way that right-handed links are incorporated, and then left-handed and right-handed and left-handed, etc., to create a chirally pure or stereopure drug that is optimized and has the chiral properties to result in the best quality drug. The important additional feature of WAVE's technology platform is that in addition to the chiral stereopure drug, oligonucleotide bases, as shown here in green, are also modified. So there's proprietary chemistry to the backbone, as well as stereochemistry and control of chirality at each junction between the nucleotides. And the result of this is that the qualities of the drug are dramatically improved, including the stability of the drug, its potency, and potentially also the duration of its effect. Now, you may say, well, this all sounds like very interesting chemistry, and the ability to produce a highly pure drug is certainly a major advance in chemistry, and the ability to rationally design the drug is a major advance. But the real question is, does it make a difference? And I think we now have very clear evidence that it does make a difference, and that WAVE's proprietary technology platform is the basis for pursuing the design of the rational design of stereopure drugs. Now, a significant advance was the publication of data relating to this chemistry in a recent issue of Nature Biotechnology. And the evidence presented in that publication, I think, is very compelling, because it helps us to understand why control over the stereochemistry, why production of a stereopure oligonucleotide drug now leads to expected improvements in the stability of the drug, the potency of the drug, and the duration of effect of the drug. In fact, the stereochemistry appears to be so important that it influences every single property of the drug, and therefore optimizing this through the process of rational design and through the incorporation of other chemical changes to the drug is very likely to result in a highly optimized oligonucleotide drug for the purpose of exon skipping. So briefly in summary and in conclusion, to summarize what I've said, Duchenne is a genetic disorder caused by mutations in the DMD gene that abolish the body's body's ability to produce dystrophin. And exon skipping over the last 15 years is a very powerful technology which allows us to correct these genetic errors and to result in the production of dystrophin protein. And the drugs that facilitate exon skipping are drugs called oligonucleotides, and traditional oligonucleotide therapies, such as those that have been developed over the last 2 decades, are composed of mixtures of drugs, of different molecules, because of the chirality within the oligonucleotide backbone leading to this drug mixture. Now, the major advance of Wave's technologies and what underpins their proprietary chemistry and stereochemistry is the ability for rational design, which allows the precise design and optimization and production of stereopure oligonucleotides. And we now know that these stereopure drugs are likely to have an impact on every single drug property, including their stability, their efficacy, and their safety, compared with stereo-random oligonucleotides. So what I'd like to do
Thank
now
you, Matthew.
is to Mike Panzara, who's going to talk about the WAVE program.
Thank you, Matthew, and thank you for everyone for attending the meeting. And I'm just going to, in the next few slides, give a little bit more specifics around our DMD program. So this slide shows the outline of what Matthew was describing in terms of how it relates specifically to the WVE-210201 compound, which is our investigational medicine for exon 51 skipping. The process is as shown here. We begin with the sequence selection, then we control the stereochemistry, looking at a wide panel of oligonucleotides, but differing the right-facing and left-facing of the RP and SP linkages to try and find optimizing towards the oligonucleotide with the highest skipping efficiency. We then look for skipping efficiency, again adjusting the chirality. We look at lead compounds. We then test those leads by applying additional chemical modifications to try and optimize. And then this led to the current investigational compound that we then took into all of our in vitro and in vivo studies. And where we got really excited about these compounds and this approach is as shown in this slide. And so let me walk you through this particular presentation. Right here on this axis, this is the percent of exon skipping that we see. And this is cells in a dish. These are myoblasts that have a deletion that is amenable to skipping of exon 51, as Matthew described. And then the bottom axis, the x-axis, this is the concentration of 210201 and a few other oligonucleotides that are used to treat these cells. And then we looked at how much drug led to how much skipping in these cells. And what you're seeing here is the blue line. You're seeing an increase in the amount of skipping that is with increasing doses or increasing concentrations of oligonucleotide delivered directly to the cells. So this is without any sort of chemicals or other things that would help the cells take up the compound, but just treating the cells in the dish, and you see these increases in skipping with increasing doses of drug. The blue compound is the one that we're bringing into trials now, and this is just on the other two lines there are one with a PMO ASO and a PS, or a phosphorothioate ASO. Now we wanted to see, that's fine in terms of generating mRNA skipping. So the big question is whether this degree of skipping, as we measured by mRNA, was going to be translating into any protein production, in other words, truncated dystrophin, and that's what's shown on this next slide. So what you're seeing here is called a Western blot. These are two different Western blots, and a Western blot is a way where you can measure the production, measure the quantities of protein. And these green bands across the top represent the amount of dystrophin measured on this slide. This shown here, you can see this is full-length dystrophin here, and then you can see shortened dystrophin products here. So what you'll see on the left-hand side of the slide, you can see the protein. It's a similar PS-ASO or a phosphorothioate ASO and a PMO ASO, and you can see that this is the compound that we're bringing into trials. These are a couple other compounds we tested, but what you can see from this portion of the slide, you're seeing a high production of dystrophin, versus this is normal muscle, so you can see a shortened product. This is, again, the Stereopure compound versus these other stereo-random compounds. Then we wanted to see, what does this look like across a variety of doses? And just similar to what we saw in terms of the previous slide with skipping efficiency on mRNA, here you're seeing with different doses of this compound, 210-201, across, you see a nice dose effect, increasing amounts of dystrophin that are produced relative to this band here, which is normal skeletal muscle. So what this led us to what we saw from this is that with this particular compound led to very high efficiency, not just at producing mRNA transcript, but that this transcript was translated into truncated dystrophin. Now, this experiment and the previous experiment were conducted in vitro, which means in myoblasts with a deletion, so in a dish, essentially. What we wanted to do is then see whether when this compound was given to an in vivo model or an animal, whether we could also see skipping. Now, there are no mouse models that enable us to do this experiment, so we decided to go directly to a non-human primate, or a monkey, where these are healthy monkeys that were treated with this sequence, because the sequence is a direct match to dystrophin in the monkeys, the dystrophin gene in the monkeys. And what was done here is that healthy monkeys were given 5 doses of 30 milligram per kilogram per week. That's about the human equivalent of a 10 milligram per kilogram per week, subcutaneously over a total of 4 weeks. We gave it to active investigational drug to the monkeys and then placebo. This is a PCR blot. This is a slightly different blot where what we did was determine whether in the monkeys could we detect skipped mRNA transcript by PCR amplification, and that's what we saw. So you can see in the control group where 210-201 was not given, there was, as expected, no skip product, yet when we gave these 5 doses sub-Q over 4 weeks, we were getting very nice skipped transcript detected in the muscles of these monkeys, and these are the muscle types: the gastroc, the tricep, the quadriceps, the diaphragm, heart, and also within the gut. So this experiment showed that what we were seeing in vitro in the cell and human cells, we were engaging target or facilitating skipping also in normal monkeys. Now, one of the things that was exciting about these results for us is that normal monkeys have intact cellular architecture in the muscles. So the muscles are completely normal. While, as many of you know, in Duchenne muscular dystrophy, the muscle cells can tend to be leaky. So when we saw that this type of transcript expression can be seen in the normal muscle, this actually was very encouraging for us and made us feel confident, based on the in vitro data and this data, that we could proceed into our human studies. So now let's talk a little bit about the trial. So the phase 1 trial is what was just initiated recently. This is a multi-center, double-blind, placebo-controlled study. This is what's known as a single ascending dose study. So there are groups of patients who will receive a single intravenous infusion and then be monitored for safety and tolerability, as well as have their blood measured for the concentration of drug. And what this does is, at each dose level, this allows us to understand how boys tolerate the medicine so that we can then plan future studies looking for efficacy. This study allows boys ages 5 to 18 who are amenable to exon 51 skipping to enter. Patients are randomized 3 to 1. What this means is that for every 4 patients who come into the study, or every 3 patients who come in the study, 1 will be on placebo. They're randomized, meaning there's a 1 in 3 chance that they're going to get that placebo. And then we're looking for boys who are ambulatory as well as those who are non-ambulatory. We're also allowing boys who've been previously treated with eteplirsen, but following an appropriate washout period. We are excluding patients who've received treatment with drisapersen, and the reason for this is there have been recent reports of some longer-term side effects of drisapersen, and we were trying to avoid any of that, preventing us from understanding the safety profile of our treatment. And of course, steroids are permitted, but patients should be on a stable regimen for at least 1 month prior to enrollment. We're looking in North America and Europe, and we expect to see data from these studies in the 3rd quarter of next year. Now, this study...
So, Mike,
yes, sure.
I'm going to stop you and ask you, could you tell us what the length of that washout period of time is if you're on eteplirsen?
Yes, we're requiring a 14-week washout period.
Okay, and the second question is, you mentioned that each boy will get a single dose IV infusion. Will, as you determine safety and you're doing your dose escalation, will those individuals be included in the next dose, or is this separate cohorts?
So the way this is going to work is that every cohort, patients who receive that single dose at any cohort will be eligible to enter this open-label extension study to receive continued treatment at that dose level. However, once we determine the appropriate dose for subsequent studies, all patients will be moved to that most appropriate dose.
Great, thank you.
Thank you. So the next slide talks just briefly about what we're looking towards the future. So where are we next? So this study, as I just mentioned, we'll have an open-label extension study. These boys will continue on treatment. We'll determine what the right doses are for the next studies. And then with a new group of patients, we'll plan to have an efficacy study. And right now, we're really in the design phases of that. We're working with the community. We're working with our regulatory authorities to design the most optimal study. Right now, we would anticipate that this would be a double-blind, placebo-controlled study, that this would have multiple doses, where we look at dystrophin expression as well as the several clinical outcomes. We would want the dystrophin will be measured the way is typical by a standardized Western blot. We would expect to do interim analyses of dystrophin expression based on muscle biopsies that are done during the study. In this particular study, we would anticipate efficacy readouts based on dystrophin in the second half of 2019. And right now we're in I mentioned this particular study we're conducting is using intravenous. This is just the first study where we're trying to get the best understanding of the pharmacokinetics and safety of intravenous. We are actively exploring both the intravenous formulation as well as subcutaneous formulations, because as I showed you in the earlier slides where we conducted studies in the non-human primate, the subcutaneous formulation was administered and yielded skipping. So that encourages us to try and also develop a formulation that we could bring forward in future trials. And finally, just an acknowledgement. We've received a lot of feedback over the year that we've been now involved. I've been at WAVE for about a little over a year now, and being a neurologist in this area who's seen this disease area evolve, it's a very exciting time. And, the idea that we are on the cusp of really an explosion of therapeutics is very exciting for me, and this is what brought me to WAVE. And the collaboration, the communication I've had over the last year with the DMD community as we've brought this program forward has likewise been very exciting and appreciated. And, this is only the beginning for WAVE. As we look forward, Wendy mentioned Exon 51. We have an Exon 53 program, but we have active research in multiple areas to try and tackle this disease because really this is our goal, is to develop transformational therapies for patients and their families. So thank you very much for your attention. I think we're going to go on now.
Yes, thank you. Thank you, Mike, and thank you, Matt, and thank you, Wendy. It's really a pleasure to learn about Stereopure chemistry, and certainly to refresh our Chemistry 101 that seems, for me at least, about a zillion years back, and I should have paid more attention, like you said, Wendy. Mike, in your presentation on slide 27, what I found really important there for this community is you, in the non-human primates, you demonstrated there that this chemistry does hit the heart. And I know that some of our discussion and our worries along the way have always been questions if we are using exon skipping and these drugs really are making dystrophin and they don't impact the heart, will the heart have more trouble? Will it be in more danger than is it built to last, essentially? So could you just refresh us about this, and is that true, or is that an assumption on my part? Do you believe that this chemistry will, in fact, treat the heart as well?
Well, the data we have is what I shared, is that in this particular, in a normal monkey, we are actually delivering oligo to the heart, and we're in those cells initiating skipping. That makes us hopeful that this chemistry does indeed help with both the distribution as well as the efficiency. And it's likely, as Matthew outlined, the stereochemistry as well as the additional chemistry that we apply to these compounds have an effect on skipping efficiency, but that also likely is affected by distribution, cell penetration. A variety of other factors are influenced by this stereochemistry and other types of chemistry that we're applying. So we're hopeful. The monkey data, normal heart muscle, that we can get in there and show skipping is very hopeful. We'll have to wait and see how it translates into the human.
Yeah, truly. We won't know until we know, but it is a very good sign for all of us who worry about the hearts of our young children. So back to the study that you described, can you inform us, or are you able to inform us about the number of individuals you'll be including in that study, the number of sites that you've identified, and if these sites that you're planning to open, and you don't have to be specific, obviously, if the contracting and the site isn't ready to go with IRB and so on, but are the sites in the U.S. and outside the U.S., and can you give us a little more framework about numbers and sites and plans?
So we haven't gone out with the number of patients or number of cohorts that we're after at this point. What we have said is that, what I can definitely say is that we're looking for several sites in the U.S. as well as several locations in Europe. So right now we are underway in the U.S. We are in the midst of site selection, and as sites are identified, they would be made public. So we would be. So I would just say that as we go along, as sites are activated, we would make that information public through. We actually have a clinical operations email account that we're happy to post that if people want information specifically like that, as sites are activated, we can get that information to them.
Great, that would be very useful. You also mentioned that individuals who had been taking the Dystrophy would not be included in this study. Will they, or are they to be included in future studies, in your view?
I would think at this stage it's a little premature to say. We don't know what the next study is going to be. I think a lot of this revolves around what we see in this initial study. I would say, though, that the age range, we would anticipate the age range of a future study potentially being a little earlier than in this later study. So that's something to keep in mind. But I would anticipate that this dry-person exclusion could be something that persists. It really is something we have to discuss after seeing the profile in the single ascending dose, as well as working with the community and our regulatory partners to figure out what the appropriate inclusion is for this next study. I think one of the things we want to be very careful of is understanding what is the profile of the compound that we have here, of the treatment, to make sure that we're not seeing complications that are not at all related to this drug but to a previous treatment. We just want to be cautious of that.
Sure, of course. Another question I think that has come up in many discussions is some girls have Duchenne, whether we call those severe manifesting carriers or X-linked inactivation. Would you consider, or have you thought about including some of those young women who are amenable to skipping exon 51 in the study?
In this particular study, we have not discussed that, and as I said, in future studies, these are the types of conversations we would like to have to understand how big of a problem this is and whether this is something we can address in a future study or by another mechanism. There are other mechanisms potentially once we see an effect in the boys potentially to provide access. Those things, I think, are things we'd have to discuss once we see the data we have.
Sure. So we just, as you might imagine, as a community, want to keep these things on your radar. And if I recall correctly, in your inclusion criteria, this did not include anyone over the age of 18, or did I not look at that slide completely? And if that is the case, then are you thinking about in the future studies with adults? I think that very often the studies end at 18, and for those of us who have young adults in our families, or for those young adults themselves who are really anxious to participate in studies, are you thinking, or could we make sure that's on your radar, the sub-studies or the consideration of treating adults?
Yeah, we've had a lot of discussions about making sure that the age is wide enough for all the reasons you point out, Pat, and that's important to us. I think what I would anticipate going forward is that, this study is up to 18. If the future studies are focused on determining dystrophin expression, I would anticipate that being in maybe an earlier patient population, but then I would also anticipate, based on the safety and what we see there, the ability to expand beyond there would be part of what we will want to try to do. The goal is to, as quickly as possible, understand if we have that the drug is safe and that we can induce skipping in a population that we'd be most amenable to that skipping, and then our definite goal and commitment would be expand from there.
Good. Thank you. As you might imagine, we will probably, I won't call it harassing, but maybe you could interpret it as harassing, because applying good drugs to people who are amenable to exon skipping technology to the entire population, is, from babies to adults, is really important to all of us, and to females as well. So we will keep mentioning it. One of the facts that we were well aware of in other studies is production, and I know you just cut the ribbon on a production facility in Massachusetts, which we are thrilled about. Does this give you and by no means am I a chemist, and people who would know me would laugh to even consider that. But in terms of production, is this scale-up production capable of really treating a community who are amenable to exon 51 skipping?
Well, the one thing that's really great about this chemistry is although it is new and it's sort of been newly developed, the scalability is quite similar to stereo-random chemistries. So we would anticipate the scale-up to be easily accomplished. Our plant here in Massachusetts certainly enables us to do what we need to do in a GMP way for all of our clinical trials, and does have the capability to be expanded to a commercial facility if it gets to that point. So we're fortunate in that the stereochemistry, when we first did it, and Matthew mentioned a paper there that we published. When we did that paper just a few years ago, the chemistry there was done a few years ago. The paper was published only recently. We were able to produce very small quantities of this, but now we're up to very large scale in a standardized way, similar to stereo-randoms, that will enable us to treat the market.
Good, thank you. And as we look into the future, and maybe this is a chemistry question for Matthew, as we think about this, in general, what we're familiar with is weekly, whether that's an injection or an infusion of an antisense oligonucleotide. So as you look at the potency and the efficiency of your chemistry, do you see that there would be those chemistries would be more effective than the burden of dosing, perhaps less burdensome to patients at the end of the day?
Yeah, I can sort of take that one, Matthew. I think that right now we're administering an IV, and simply for the single ascending dose study, that's going to tell us a lot about the metabolism or the concentrations of the drug. If we have a high impact on the production of dystrophin, we're very optimistic that that dystrophin production, with a high level of dystrophin production, we should be able to back off on the frequency, theoretically, as required to keep that production level high enough. And that's the other reason we're looking at subcutaneous, is that if we can develop a subcutaneous formulation that is well tolerated, given the substantial differences in the chemistry between this compound and drisapersen, we're optimistic that we can develop another formulation that then allows us to give flexibility to enable, even if it has to be more frequent, to make it less burdensome. The chemistry and the effect that's happening should enable us to do that, but we won't know until we see the preliminary information.
Thank you. So just as a practical question, what is the turnover dystrophin in terms of length of time?
That one I'll turn over to Matthew, because he obviously is the expert in dystrophin production.
Yeah, Pat, I think the answer is it depends. I mean, the turnover of dystrophin protein is very slow, so it's a large structural protein. So we know the turnover takes weeks and probably many weeks. It could be up to 15, 20 weeks. But I think what we don't necessarily know is about the turnover of abnormal dystrophins. So, for example, when you generate an exon-skipped dystrophin, it's not entirely normal. And whether that will have the same turnover rate as normal dystrophin, or whether the turnover rate of dystrophin in different muscle groups or even the heart muscle will be the same, I don't think we really know. But the turnover is certainly, even of exon-skipped dystrophins, likely to be very slow and many weeks, probably in excess of 10 weeks.
Good, thank you. Back to you, Mike. One question about biopsies. As you know, we as a community have had our share of biopsies and our experience with biopsies. You're talking about a single infusion. I'm making an assumption, although please correct me, if these boys will have a baseline biopsy and then another biopsy in the future. Can you describe that, or have you really nailed that down about what we might expect?
Sure, I can describe that. one thing we've heard an enormous amount of feedback on are biopsies. We've heard feedback from patient community, from the physician community about where to biopsy, when to biopsy, how often, and that biopsies are a serious thing that we shouldn't overdo. And so with that in mind, in the single-dose study, there is no biopsy for entry. We are not doing a biopsy as part of this study because the focus is safety, tolerability, and drug concentration. However, as I mentioned, there's an open-label extension study. So after we have a period of time after that one dose where we allow the drug to clear, I said we would be offering active compound, investigation compound 210201 in the extension study. Prior to starting multiple doses of that, we are requiring a biopsy at that baseline because we want to, before now giving potentially therapeutic doses over time, to have that baseline biopsy. And then we would anticipate one additional biopsy as part of that open label biopsy, potentially, we're discussing now 6 months to a year down the road. Just to
Great, thank you.
Expression. Yes.
Great, thank you. I so appreciate that. Now we're actually near at our 2 o'clock or 1 hour time period. But I want to ask you, and this is a very forward question, and it's future and just how you think about your technology and this unmet need. There are, obviously a need for skipping in the very early exons. And multiple skipping of one or more exons. So as you think about, and Matthew, this is probably a question for you, as you think about the practicality and the possibility of skipping exons early in the gene and multiple exons, are you thinking about that? Are you doing research to understand that? And if so, could we update at some point and could we understand it better? Because as you know, some of these young guys need 2 exons skipped, and certainly there is a need across the full length of the exons and dystrophin to get some skipping done. So what do you think, if you close your eyes and think about the future, can you help us think through that with you as well?
Yes, so I think, shall I
go ahead, Matthew? Go ahead, Matthew, yeah.
I think, for a long time, the scientific community has just been focused on trying to make an exon skipping drug that works. And to some extent, the scientific community has got there with the breakthrough that Sarepta made with eteplirsen. But I think it's really, these problems that you've described, particularly the multi-skipping or the requirement to skip more than one exon, and even the desirability of skipping multiple exons so that a single drug could treat many, many patients, is something that's been talked about. But I think, A, the focus has been elsewhere, and B, there hasn't really been the technology to facilitate this. So I think one of the really exciting things about WAVE's technology is going to be the ability to think about these harder problems, because the ability to really design the oligonucleotide drugs in a rational way, to sift through many, many stereochemical versions of the drug to find the one that is perhaps the optimal one for skipping multiple exons, or skipping more than one exon, is really exciting. So I'm not sure. Although there have been some efforts made to tackle these questions in the past, there haven't been very serious efforts, I would say. But I think, with this type of technology, the scientific community is now in a position to think seriously about this, and it doesn't mean that we'll solve the problem or solve it necessarily within a short time. But I think, with this technology, it gives us a chance of really trying to understand how to optimize the oligonucleotide drug for the purpose of skipping one or more exons. And so I think, the future in the next 5 years, I would guess, is going to result in progress along these lines, multi-exon skipping drugs, as well as really understanding how to optimize improved drugs of the kind that WAVE is developing, and potentially incorporating ways in the future to allow these to be delivered even more efficiently to the tissues that we're interested in, the skeletal muscle tissues, but also the heart muscle and the nervous system. So that ideally in the future, one would have a drug stereochemically optimized, a stereopure drug that skips exons very efficiently, that gets delivered very efficiently, and that could even be deployed to skip multiple exons. And I don't think it's fanciful to think that we could be there in 5 years.
Thank you. So I wanted to wrap this up by saying thank you to Wendy and thank you to Matthew and thank you to Mike. We are delighted with a very specific targeted chemistry. We look forward to your progress and Matthew, so that's a desire and a wish on our part to be able to treat as many children as possible and as fast as possible. So we are very thankful for Wave Life Sciences in our space. We look forward to the study. We're thankful for your thoughtful approach to this study, and we wish you Godspeed and hurry up because this is really needed. So thank you all for joining us today, and we look forward to an update very soon.
Thank you very much for the opportunity.
Thank you, Pat.
You're welcome. Thanks, Wendy. Thanks, Matthew. Thanks, Mike.
Thank you.